So should we go ahead and get started?
Sounds good.
Okay, so please interrupt me at any time whatsoever. I do not have this well organized as usually. I do not have this well organized, but we're gonna plow through it. So this is the paper that we're doing today for the Aspirational Neuroscience Journal Club. It's called Behavioral Timescale Synaptic Plasticity Underlies CA1 Place Fields by Bittner et al. And this is a paper that I've actually been meaning to do for a while. When I came across this idea, actually in a lunch conversation with the last author many years ago, it seemed like a very, very relevant counterexample to regular Hebbian plasticity. So let's go along the lines of how this paper is setting it up. So this paper is setting it up saying, look, there's this thing called Hebbian plasticity that has been studied enormously in hippocampal slices, etc., and it has a very particular role. It says it has two core elements. The two core elements of Hebb's postulate are causality and repetition. If the activity in a presynaptic neuron repeatedly or persistently takes part in the firing of a postsynaptic neuron, then the strength of their connection should be increased. So it's essentially saying, look, if there's a dendritic spine synapse onto a postsynaptic neuron, and that postsynaptic neuron is having an action potential. So its voltage, its membrane voltage is elevated, and at the same time, or very close, within tens of milliseconds, there is a presynaptic action potential that causes the presynaptic side of the synapse to secrete glutamate. Then you're going to get the classic long-term potentiation, where that glutamate is binding not only to AMPA receptors but to NMDA receptors. The NMDA receptors are usually would allow calcium into the dendritic spine, but that the NMDA receptors are blocked by magnesium ions unless the postsynaptic cell is elevated in its voltage, like it had just fired and had a let's say a dendritic back propagating action potential. And so the NMDA receptor itself is acting as the coincident detector. And that coincident detector is saying, hey, I've got to have a postsynaptic action potential, and I have to have presynaptic glutamate. And if that occurs, then the NMDA receptor allows calcium to flow into that particular synapse, and that particular synapse then has downstream activation, maybe of CAM kinase 2, etc., that will cause that synapse to grow and strengthen in its strength. The key idea behind that is, for this paper's point of view, is that coincident activity is really happening at the tens of milliseconds or less scale. I think they throw out a number like about 50 milliseconds. They argue in here. So what they show in this paper pretty convincingly, and this is a 2017 paper, I believe, and it has been, as far as I've seen, replicated multiple times and factored into the literature in a pretty accepting way. So I don't think that there's I don't think this is wrong. I think the results here should be accepted. So what they show here is a totally different type of synaptic plasticity, what they call behavioral timescale plasticity, where the basic assumption of Hebb's rule is no longer the case. The presynaptic activity does not have to cause or even come close to causing the postsynaptic activity. It doesn't have to be within a few tens of milliseconds. It doesn't have to be repeated. It is something that will strengthen a neuron, strengthen a synaptic connection onto a neuron that has had what is called a plateau potential. And anything within about 5 seconds, minus 3 seconds to plus 2 seconds, I think, around that little plateau potential in the target neuron, any synapses that have been active within that time period will be strengthened, and they show that. So let's go ahead and go through the paper figure at a time. So here's figure 1. So what they're doing here is they're doing hippocampal place cells. They're recording from CA1 area of the hippocampus. Let me show a particularly important figure, not in this paper. This paper doesn't have any diagrams of the hippocampus or anything, unfortunately. So what we have here is we have CA3. We have the hippocampus. We have CA3 that has place cells in it. It has neurons that are tuned to the place where the animal is. In this case, the animal is a mouse and it's running on a track. And so the idea is that this linear track has a few features on it and stuff. And so the mouse will make a cognitive map of where it is on that track, on that linear track. And there will be particular CA3 cells that will respond to each location. This is the assumption, each location on that track. And then those CA3 cells will synapse onto CA1. And CA1 also produces place cells, cells with place fields that will respond to particular portions of the track. But it is known in the literature that CA1 has more flexible, I think is the right word, more flexible place fields. They often pop into existence very suddenly during trials. So you'll get a cell that was silent, and then all of a sudden, bam, it will become a place cell, and it will be a place cell for the rest of the experiment. And what they're doing is they're looking for how that phenomenon occurs. Now, the key thing about these CA1 cells is they get inputs from CA3, and those are the synapses that are presumed to be being strengthened. So their place field is going to be presumably made up of place fields of the presynaptic CA3 cells that are synapsing on it. And in order to create a place field in the CA1 cell, you just need to strengthen the right synapses from CA3. There is another input to these CA1 cells on the very apical tufts of this. It's here in the diagram. These apical tufts are having synapses coming from I believe the entorhinal cortex or other cortical areas. And the idea is that those are actually providing the training signal here. That they are, although this paper doesn't demonstrate that, other papers do. The idea, though, is that these plateau potentials are actually caused by cortical training signals from outside. Okay, so here's an example of an experiment. We've got this this mouse that is running along a treadmill. The treadmill position is shown here on the x-axis. It goes from 0 to 200 centimeters, so it just goes around in a circle. And the laps that the mouse has gone are shown on the y-axis here. And so for the first 10 laps here, nothing. This particular cell. This is a recording of a particular cell in CA1. It's a intracellular recording of a particular cell in CA1. It's showing the firing frequency. 25 hertz is when it's in red, down to blue means 0 hertz. It's showing the firing frequency of that CA1 cell at different points on the track. And for the first 10 laps or so, that particular cell is silent. And then there is something called a plateau potential. So it's shown here as happening at about 110 centimeters on the track, that position. And so if I look at figure B here, this is the membrane voltage of this CA1 cell on the laps prior to this plateau potential. And then on a lap 11, for some reason, out of the blue, presumably because of inputs from the cortex onto the apical tuft dendrites, there is a little less than a second long plateau potential. Now, what is a plateau potential? A plateau potential is when there is usually when there's an action potential, the membrane voltage goes up, and then it very quickly comes back down within a few milliseconds because potassium currents, potassium channels open up to drag that membrane voltage back down. For whatever reason, in a plateau potential, that does not happen. There are other ion channels that are very much involved with this. And crucially, some of them, although I'm not sure this is causal, but some of them are voltage-gated calcium channels. And these voltage-gated calcium channels are going to not only keep the membrane voltage high, but it's going to have a lot of calcium allowed into the cell during that particular time period. So in any case, that's a plateau potential. That's kind of the training signal here. It just comes out of nowhere. And then from then on, this particular cell is responding to these areas of the track for the next 30 laps that this mouse runs. And if we look at lap 27 here, this is what it looks like as the mouse is going through this area. There'll be a whole bunch of spikes. Those spikes are action potentials showing that there's increased firing rate for this particular CA1 cell, which means that it has a place field at that particular location. Any questions so far? Yes?
Yeah, hi. Do they tell us that how many laps the plateau potential shows up? Is it just one lap and then the learning is done and then there's no more plateau potential, or does the plateau potential also show up for a few laps?
Yeah, that's a great question. The assumption is definitely that this can happen on one lap. This is a one-time phenomenon, but they actually give a thing here. that I was having a bit of a hard time understanding. Here we go. The average number of trial seems to be the exact same thing as lap. So trial and lap are the same thing. Please, anybody can correct me if I'm wrong about this. If you've read the paper and I'm just butchering it, please step in. So the average number of trials containing plateau potentials during the induction phase was 1.4 for naturally occurring plateau potentials. So essentially, sometimes it happens twice, but a lot of times it's just one for a naturally occurring plateau potential. That means that it happened out of the blue. They don't know why. And 5.1 for experimentally induced place fields. So they also have the option of experimentally injecting current to cause a plateau potential. And how I'm reading this is that when they do that, they do it for about 5 laps, all at the same position.
Ken,
yes.
the out of the blue aspect really bothers me because, why does it happen at one point and not at others? Why does it only happen here during encoding and not some later retrieval? If it happened during retrieval as well, it could easily mess things up. So, yeah, that aspect, that detail.
Yeah. What do you mean by retrieval?
When you're not, retrieval. When you're re-experiencing, when you're using your learning in order to, for example, find a direction or something like that. If you're applying this in an experiment where you have to use the episodic sort of overlap between place fields that you've learned.
Yeah.
Yeah.
So how I'm interpreting this is that from a behaviorally useful perspective, you might want to generate a few more place fields in areas that you would like to cover with memories and to map, right? And so it would make sense if there is some mechanism, some top-down controllable mechanism that can say, hey, I want a few more place fields in this region right here at this exact location. And maybe that would occur because there aren't enough place fields to tile this area that you're exploring. They actually show that if the animal kind of slows down in an area that while it's learning, that it generates more place fields. Not in this paper, but in a different talk that I was looking at from the authors. So I think out of the blue doesn't really cover it. Out of the blue means there's some top-down mechanism that we don't understand that is causing this. And yeah, but it is one-shot learning. All right, so one of the things that's a little bit confusing about the paper, or at least I found confusing about the paper, is they keep talking about the voltage ramp that is occurring in these neurons, and basically all that is if you look at this trace over here. Oops, sorry. If you look at this lap 27 place. It's firing at a particular location. That's the location that it's representing. That's its place field. But you can see that at a sub-threshold level, if I look at the membrane voltage, the membrane voltage has a little hump in it here. And that's what they're calling a voltage ramp. And they're using this as a proxy to say what is the actual synaptic inputs to this cell. And if you look at lap 10 before this learning event occurred versus lap 27 after it had occurred, there's no bump in lap 10 anywhere, and in lap 27 there's a bump. and that bump is essentially used synonymously with the receptive field, the place field location, if they map that onto the positions of the track. So in figure C, 1C here, that's what they're showing. So here's a particular neuron. They've gotten rid of the action potentials. They've thrown those away and then did a low pass filtering. Okay, the action potentials are there, but that's not the really thing that's important. They want to know kind of what's on the input synaptic side, and so they see this ramp of activity, this bump of activity that's going from, brings the neuron from minus 60 to minus 52 millivolts. These are sub-threshold, but it's saying that there's synaptic inputs that are coming when the mouse is running through this area of the track. And these ramps are always roughly centered on where the calcium plateau, the potential occurred at the previous training lab. And some of the things that so they see this a lot. They show this around, and one of the things that they predict from this is that if they have a bit of a modeling here that I don't want to go into too deeply, but they have some assumptions that they make to say, look, if Hebbian plasticity was the only thing that was causing this, then what would we expect the width of this place field to be? What would we expect the width of this ramp, this VM ramp, to be? And first of all, they suggested it would essentially exist because there are the place fields themselves on CA3 on the input are extended in space. They're extended in space, and you can see this in figure E, right? So figure E is kind of showing what you would expect from the standard Hebbian plasticity. Figure F is what you would expect from this behavioral this long-term, long-scale plasticity that they're showing. And essentially, if there's there would be an extended place field on this CA1, even if the synapses, only those synapses that were active during the very short, less than a second plateau potential were strengthened, because the CA3 place fields are extended in space. And so what you might get is you might get something that looks like a bump that's extended. But if the mouse was running more quickly through the maze while that calcium potential occurred, only during that particular trial where the calcium potential occurred, it wouldn't matter to the width of this voltage ramp. And the reason is that the calcium plateau is so short that it's essentially a snapshot of all the CA3 cells, their place fields, that were active during that snapshot, right? Whereas for a long time scale plasticity, where the presumption is that you actually are strengthening synapses that were occurring within a few seconds, as opposed to 50 milliseconds, within, let's say, 5 seconds of the time of this calcium plateau. Then, if the mouse was running faster on the track, it would have, within those 5 seconds, actually covered more different places on the track, and all of those different places would be strengthened. And if you look at figure G, that's exactly what they're showing. That's what they're seeing. A standard Hebbian plasticity where the time period that's strengthening things is 50 milliseconds would show no dependence of the ramp width versus the velocity of the mouse during that calcium plateau trial. And what they see is they see a nice linear trend where if the mouse is going more quickly, the width is larger. Let me say something about these figures. These figures are actually these figures up here are more properly explained over here. So here is a mouse running, figure 2A here, mouse running. The idea is that there's a bunch of CA3 place cells that we are not seeing. Sorry about this. We're not seeing, but we're assuming that they exist and are impinging on our CA1 cell. The heights of all of these are really making the assumption that, hey, there's a random number of synapses from each of these cells onto this particular CA1 cell, and any of them could be strengthened if we desired, if there was a plateau potential at any place on the track. And then after there is a plateau potential, some of these CA3 cells get strengthened, and that's what the height of these Gaussians are showing here. That they are that the CA3 cells that were active closest to when the plateau potential occurred are going to be strengthened the most. And if they get strengthened in this way, you're going to get a V ramp that looks like this. I'm going to pause for questions. Anybody got a question on this?
That makes sense to me.
Okay. Okay, great. So now what figure 2 is doing here is they're creating a model of this. They're saying, okay, let's assume that this really that this behavioral timescale plasticity is really occurring. How we're modeling this is we're going to assume that there's a set of CA3 firing rates coming in, and I'm going to switch over to this is their supplementary material. And so they have a model of this, a mathematical model that is supposed to capture this. So this is the new weight that is going to occur, the strengthened synaptic weight from CA3 cells that are responding to position Z on the track. Okay. That's what this, so it's basically saying, hey, how is the synaptic weights from place cells in CA3 at position Z on the track going to change? And they suggest it's going to be the existing weight plus this learning rule. And this learning rule says there's a time period of the CA1, this particular CA1 cell's plateau potential. Okay, it only lasts for about 300 milliseconds, so this plateau potential is very short in time. It is going to be convolved with the CA3 cells that were active. That this particular CA3 cell that was active at any of these times that are being convolved. And that's going to be dependent upon where the mouse is, but it's also going to be dependent upon the trajectory of the mouse as it's moving over this, let's say, 5-second period of time. And then this K is actually the learning rule that they're proposing. And so this K is saying, given that the plateau potential is happening at, I think the idea of the plateau potential happens at T prime, I believe. So if the plateau potential happens at T prime, this K function is going to say how much strengthening occurs a second before that or a second after that, and vice versa. And so what they do is they take the trajectory of the mouse that they're recording. They take the V ramps that they average from the trials after the plateau potential that really says what the how this CA1 cell's receptive field has changed. Okay, and they put that into a big complicated math solver of some sort that I can't understand, and they determined this K function for that. And that is what they're getting here with this plasticity rule. So this plasticity rule is this K function that they're fitting, and they do that with all their data. They get all of these plasticity rules here, and this is the average plasticity rule that they find. And if you look here, the time from induction, 0 is when the plateau potential occurred. It goes all the way from -2 seconds and beyond to +2 seconds. So it's about, it's maybe goes from -3 seconds to +2 seconds around this plateau potential. Now let's look at this from the point of view of a single synapse. So that's what F is doing here. So figure F here is showing from the point of view of a single spine on this CA1 cell. So this is a spine's point of view. This spine is getting input from CA3. Okay, it's getting a bunch of these are all sub-threshold. If you look, this is a 10 millivolts. These are a little. This spine voltage is these are excitatory postsynaptic potentials, right? So the presynaptic action potentials shown in gray here, these little gray dots, they are creating release events. This is a biophysical model, by the way. These are models that they have. That creates little excitatory postsynaptic potentials, which means there's glutamate that is being squirted onto this dendritic spine. Now, here is the key thing that they're assuming here. They're saying, in our model, these excitatory, these glutamate events are going to create a local signal that is shown in black here, that is being integrated. That local signal is something to do with the molecular basis in that particular spine. It is what do they call it? It is an eligibility trace. Most likely it has something to do with the, not in this paper, but in other papers I've been reading about this, most likely it has something to do with metabotropic glutamate receptors. It's a long time scale, multiple seconds, local signal to this synapse that says, hey, this synapse has been active in the last few seconds. So that's this black line here. So I want you to think of that as that synapse is storing molecularly that it has been active in the last few seconds. The idea is that a plateau potential then comes over the entire cell. That plateau potential gives a global signal that is lasts for a certain amount of time that is shown here in blue. This may be simply just calcium influx. It could be as simple as that. And And each synapse is essentially asking the question, what is my local signal, this local signal that is my eligibility trace that really says, have I, as a synapse, fired in the last few seconds? What is the overlap between it and this global signal that's coming from the plateau potential? And to the extent that there is an area of overlap, which is shown in this gray shaded region of the two, that is the amount that the synapse will be strengthened. That is behavioral timescale plasticity in a nutshell right there. Any questions on that?
No, not about the model itself and how it determines the weight change. I mean, this is a good representation of what they find, and this kind of this makes sense to me. I'm still just mostly wondering about the initiation of these ramps. Yeah.
So let me. There's only one more figure left in this. So let me go through that figure, because this figure basically takes away all of the assumptions that are made of the behaving animal, because they go to a slice model. So they take hippocampal slices from an adult mouse, and they're doing electrophysiology on those. They remove the area CA3, so there is no input to these CA1 cells, but they do electrical stimulation of the stratum radiatum axons, the CA3 cells that would have the axons from those CA3 cells after they were cut off. They do electrical stimulation of those to simulate CA3 input. And here's the experiment that they do. So they've got this hippocampal slice, right? They've cut off all the main inputs. They've got a intracellular electrode into one CA1 cell. They are going to give it a plateau potential. So they're going to inject current into the soma to give it a plateau potential, and that's what this is showing here in B. But they also are giving, through electrical stimulation of the axons going into this synapsing onto this CA1 cell, they give some action potentials before they do this plateau potential training event to get a baseline. That's what's shown in black here. And then they do the plateau potential when they're firing these electrical potentials at different times relative to this plateau. So for instance, these are 10 action potentials that are fired in at 750 milliseconds before the plateau potential. These are 10 that are fired in at minus 250 milliseconds. These are 10 that are fired in at 750 milliseconds after the plateau potential. Yes?
Sorry if this is a dumb question, but they've just checked prior to this that there's like a monosynaptic connection between those axons that are cut off from the CA3 onto these CA1 cells. So like they've already confirmed the two are connected, and then they start this pulsing. Is that right? Or
I think yes. In the sense that they are measuring excitatory postsynaptic potentials in the CA1 cell before they do the plateau potential.
Yeah, great, thanks.
Okay, and so if we look in, so the experiment would give a plateau potential. It would measure the excitatory postsynaptic potential for a particular excitation before. That is being put at a relative amplitude of 1. Okay, so it has a 1 here. And then if they give these 10 trains, 10 pulses, 750 milliseconds before the plateau potential, and then test for many minutes afterwards, they get, in this case, a factor of 2 increase. If they do it a little bit closer in time to the plateau potential, they get a factor of 3 increase. And it's a long-lasting increase in synaptic strength. And if they do it after the plateau potential, they get about 1.5 in time increase. And they did this at many different times relative to the plateau potential, so that they could plot out the entire learning curve. And this learning curve is exactly what in these previous things they were plotting here. So this is the behavioral timescale plasticity learning curve. Anything that happens 2 seconds, 2 full seconds before the plateau potential, any synapses that are activated even 2 seconds before get 1.5 amplitude increase in their synaptic strength. Anything that's happening exactly at the plateau potential gets up to 3 times the strength. Anything that's happening 2 seconds after gets a little bit of strengthening as well. That any questions on that? So to me, this yeah, go ahead.
This perhaps gets into the discussion, so we can pause this for a moment. But I'm just trying to think conceptually, is this really so different from Hebbian plasticity, just with a much longer time scale than we're used to for Hebbian plasticity? Or is it actually, yeah, like fundamentally completely not at all appropriate to call this Hebbian with a long time scale?
Yeah, I mean, that is a great question. I feel like they did a little bit maybe of a straw man at the beginning, where they said this is Hebbian and we found something that's not Hebbian, whereas I suspect there are a lot of readers that would say,
but it depends on, doesn't it depend on where the plateau potential originally comes from, like why it happens, what causes it to emerge? Because, you know, if that emerges because of postsynaptic activation, then you could say, yeah, this is another form of long Hebbian plasticity. But if it is induced by something else, so if this is, you know, there's another top-down mechanism causing that to pop up occasionally, then it could be something unique, something different.
Well, I think under their definition of Hebbian, which is the cause of the firing, anything that is essentially participating in the cause of the firing of the postsynaptic cell should be strengthened. This would definitely be well outside what anybody would call Hebbian, regular Hebbian plasticity. I think I think Ariel is right, though, to say this is kind of a stretch of what we would normally think of as Hebbian plasticity, but it's not radically different. And there are a lot of times where that we've seen in other papers where there's something that we're calling Hebbian plasticity, but the quote-unquote cause is on another pathway. So if you look at the amygdala fear studies that we've been covering, the firing of those cells during the plasticity event was essentially from the fearful event itself, not from the auditory stimulus, let's say. And yet those auditory axons their synapses were strengthened. Yep, go ahead.
Yeah, I mean, in terms of comparing to Hebbian plasticity, I think that sort of brought up a question in my mind. Does this learning occur? So it sounds like the action potential is what they're focusing on, but what if the cell does not fire? So is this dependent only on the action potential, or does the cell have to fire? Because in Hebbian, you assume the cell fires.
Right.
Well, I want to be careful, because there's another thing that's very important here, and this is a plateau potential, and so regular Hebbian plasticity, it's just action potentials, general action potentials, right? This is only occurring with the plateau potential, and this is a one-shot plateau potential that can produce this type of, CA1 cell that has a place field, whereas that particular cell did not have a place field before.
But do you have to see postsynaptic action potentials occurring on the plateau for this to happen? Or can there be a plateau with no postsynaptic spikes riding on top of it?
I have no idea. I assume that it is hard to generate a plateau potential that does not have action potentials that are occurring.
Right.
I would assume that I would assume that all you need is calcium. Actually, the final figure. I'm sorry, I lied. There's one more figure. The final figure may have something to say on this, because what they did is they blocked they blocked NMDA receptors. That's in this DAPV that they put in. They blocked NMDA receptors, or they blocked L-type calcium channels, and they showed that both of those interventions decreased this behavioral timescale plasticity if it was blocked during the plateau potential. And I think what that generally means is that you need a calcium influx. That's how I'm reading it. It's like so the real thing that's happening is not the action potentials or anything like that. You need a sustained calcium flux into the cell that is going to be the global this global signal that all of the activated synapses feel to order to say, hey, I have an eligibility trace. I have been activated by glutamate within the last few seconds. So if I see a big calcium flux coming in from the global stuff, I'm going to strengthen.
But Ken, so if we're still talking about Ariel's question in a sense, and I am interested in that question, then okay, an MDA is required, calcium influx required, not a problem. This is all standard STTP stuff. Just because, so if we just want to look at it as a rule, what fires together wires together, even if the plateau is being caused by something else, say entorhinal cortex layer 3 or whatever on the tuft, somehow causing the plateau to emerge, and therefore you've got calcium influx, and therefore you can achieve this kernel for weight change. As long as that still causes the postsynaptic cell to also fire because the plateau is there, then essentially, just from a rule perspective, you still have what fires together wires together. It's just that you are causing this plateau in another way, and hence you're getting the postsynaptic firing going on for a nice range of time.
And the firing together in this case could be
extended over time. Yeah, the window is big. Yeah.
I'm just checking conceptually, even if it was, let's say, altered intrinsic plasticity in the postsynaptic neuron, like it was just way more excitable for its own sake. But it was still
I mean,
developing.
That wouldn't explain why it's a place field, though, right?
No. I mean, in general, like in a different, just in a conceptual sense. If you had a postsynaptic neuron that, for whatever reason, spontaneously was more excitable, but it still went through, it fired after receiving a presynaptic input, and that connection still strengthened, like we see here, we'd still call that Hebbian learning, right? I'm just trying to work out what's an example of like clearly non-Hebbian sort of strengthening that would be
I would consider it truly non-Hebbian if you got strengthening when there was no postsynaptic firing happening at all. So if you could show that you just got the uptick, the ramp, the plateau, and so calcium influx because NMDA is activated, is opened, the block is removed a little bit, and then you're getting strengthening, and you didn't even have to see a postsynaptic activation. In that case, that would be fully non-Hebbian, right?
Yeah, that makes sense. Thanks for clarifying.
Well, at least that's my take on it. I'm not saying that that's how you should interpret it, but that's, if we're just looking at it as a rule, yeah.
I want to point out one thing. This is a little off topic, but I forgot to say this. So there's this paper from 1993 on a completely different non-hippocampal place cell topic from Edmund Rolls et al. And what they propose in this paper is that the way that human beings learn to do translation invariant and view invariant object recognition, visual object recognition, is something called the trace rule, where somehow, some way, neurons in this case higher visual areas, not only strengthen their synapses due to the Hebbian rule, but they strengthen their synapses over an extended period of time, so that when the baby, let's say, is looking at a translating, rotating object, that all of those different inputs are strengthened onto the same postsynaptic cell, and that cell becomes able to recognize that object under many different views. They put this forward as the trace rule. They have a model that shows that it works and everything. And this is just kind of an example. I don't know if anybody has actually put this together with behavioral timescale plasticity, but it's an example of, even if you go back in the old literature, they're talking about things that are not classic Hebbian needing to be brought in to explain things like view invariance in object recognition. So I think this particular paper can go a lot farther than just this particular hippocampal place field stuff.
So what's this got to do with memory decoding?
Well, so
encoding is fun too. Okay.
So place fields are potentially an excellent place to want to do decoding. So if you think about it, I'm speaking off the top of my head here, okay, but presumably there are a bunch of CA3 cells that could have their receptive fields measured in an experiment, maybe calcium images or something, and CA1 cells that could potentially even be caused to have very particular place fields by this technique. In fact, there's a paper here that has I won't show it, but there's a much more recent paper that does an all optical electrophysiology with voltage sensing and stuff that actually replicates all this stuff and induces place fields with these plateau potentials all optically. You could imagine an experiment like that where then you do connectomics afterwards and say, hey, can we decode the place fields from the synaptic strengthening onto these CA1 cells that have also been decoded? So that's
can you elaborate a little bit? So then if you were to use, say, some kind of microscopy or whatever to decode the place fields, how does the fact that BTSP was involved in creating those place fields change anything about decoding the place fields?
I don't think it does, actually, but let me bring up one example of that. So you had the paper with the Sarah Blockface imaging, UTEPO. What was that paper?
Yeah.
Yeah.
I don't know if it's pronounced UTEPO, but yeah, sure.
And it actually was looking for ingram to ingram and didn't find it. I throw out the possibility. I'm not sure if this is the case or not, but I throw out the possibility that a different learning rule, something like this, might actually make a disconnection between. It might explain the lack of connection there. I don't think it would, but it's a possibility. So in any case, the larger answer is behavioral timescale plasticity probably does not change any of the main decoding ideas. It really is still, we're looking at synapses onto postsynaptic cells, and the postsynaptic cells receptive field is supposed to be predicted based upon the strengths of synaptic connections from other cells that have their own receptive fields. That's the kind of classic, basic, minimal memory decoding experiment as we think of it. And so the fact that behavioral time scale plasticity might have generated those synaptic strengthenings probably is irrelevant.
Yeah, but if we move from pure memory decoding to the problem of structure to function, then suddenly this does have a significant impact.
Yes.
Yeah.
One other thing I guess that's relevant here is I'm curious about how long this strengthening induced by behavioral time scale plasticity lasts. If you come back, because the thing about place cells, right, is like they can be quite stable. And if you come back the next day or a few days later, you can still see these same place fields and put the animal back in the same environment. But I would be curious as to, like, do you see the same effects at those later time points? And does the maintenance of those same effects require some structural changes in the meantime in the animal's brains? Or somehow this BTSP still kept through purely like phosphorylation or other sort of non-structural changes?
It's a great question. Randall may have more information than me on this, but I did a quick look, and a quick Google search seemed to suggest that CA3 place fields are much more long-term, and CA1 place fields are more transitory, or at least they can pop into existence. Maybe they pop out of existence just as easily. It's a good question. I've been assuming that it's the same, but very well, given that this is in the hippocampus, it probably doesn't last for forever.
Yeah.
Yes. Go ahead.
Have they been able to see this BTSP behavior in other parts of the hippocampus or other types of cells? Has anyone tried and failed or succeeded?
I don't know. it's a great question, and I didn't even look it up. I only saw references to hippocampus CA1. Anybody? Did you know if it's been seen anywhere else?
No, I have not seen it in any papers that are not about hippocampus so far.
There's a Jeff Magee and somebody else. They did a review article in, I think, 2020 that covered just general synaptic. It had a lot of stuff about behavioral timescale plasticity, but it was comparing it to all the other different types of regular Hebbian and three-factor, with dopamine and things like that. I suspect that might have a better review of the literature around that question.
Thanks.
Because they were definitely arguing that this is not just a thing in the hippocampus, that this could be a general thing that did one-shot memory encoding everywhere where you needed to do one-shot memory encoding over a behavioral time scale, like 5 seconds.
Yeah, I think that would be very interesting, because I think I saw a reference somewhere where Jeff Magee referred to this plateau as equivalent of a Brack propagation that sort of facilitates learning. So yeah, it would be good to see if this is more general.
Yeah, I think in general the aspirational neuroscience community, we need to kind of bone up on some of these other non-standard but still well-researched memory models, even if they're not immediately obviously connected to memory decoding.
I would agree. Yeah.
Yeah.
I know we're over time. Any other questions?
I have so many questions, but I don't think we can answer them right now. Yeah.
You want to give a plug for the next paper, or do we have a next paper?
We do have a next paper, don't we?
I believe Randall is given the next one, if that's still the case, on the cryo EM from memory, or have we switched? I can pull it up.
Yeah, so I had switched that one out before. I might still want to do that one unless I find a different paper that I prefer to do. But what may change, just because of my having to be in the Netherlands right now for a family thing that's, a bit of an emergency thing I had to attend to. I'm not 100% sure if I want to be the next one to do the presentation. It is possible that it's fine, because it looks like I'm heading back to the US probably in the second week of September. But I want to be sure that everything just works out time-wise. So I'm gonna follow up on that.
Okay.
How about
if you need me to swap in for you, I can do that.
Yeah, I was going to say
maybe it's safer if we just do that. If we assume that we swap. Yeah.
It's been a little while since I've done one, so let's just say I'll do the next one. Unless anyone else here wants to have a go, everyone's welcome.
Yeah.
Nah.
Go ahead, everyone.
Well, you don't have to sign up right now. If anyone else who's here would like to give one at some point, then you can email and we'll sign you up as well. Unless you do want to sign up right now, but otherwise I'll assume I'm doing the next one.
Great.
Awesome. Thanks.
Thank you.
Thanks, Ken.
That was super intriguing, and leaves me with so much more to think about and research and read about.
See everyone in a couple of weeks.
All right.
Bye.
Bye-bye.